Hedrin was first launched, its unique patented 4% dimeticone formulation and its brand new way of killing lice without pesticides heralded a wave of products which claimed to kill lice in a similar way. Some of these products claimed that they suffocated lice, but their mode of action had not been demonstrated.
We always believed that it was highly unlikely that 4% dimeticone caused asphyxiation in lice. Independent commentators agree that the anatomy of the spiracles and tracheae make it virtually impossible for suffocation to occur. Lice can survive oxygen deprivation for many hours, and in any case, silicone oils let oxygen through. So to confirm exactly how 4% dimeticone did kill lice, we needed a thorough investigation of exactly what happened under experimental conditions.
The experiments were carried out by a team in Cambridge University's Cavendish Laboratories, using electron microscopy to demonstrate how Hedrin's active ingredient dimeticone penetrated into the louse. Live adult head lice were collected from volunteers. Half the lice were treated by coating with Hedrin and left overnight at room temperature, with the untreated lice used as a control group. Both treated and untreated specimens were prepared for electron microscope examination, as the first stage in detecting whether dimeticone from Hedrin had penetrated the spiracles lice use to take in oxygen and to pass out water vapour. A more detailed chemical examination was then carried out, cutting tissue away with an ion beam and using X-ray microanalysis to detect the element silicon inside the spiracle and trachea. Silicon is present in dimeticone but not naturally present in lice, so the technique was a very effective way of showing how far inside the louse Hedrin had penetrated.
The results of the experiment were extremely interesting. X-ray analysis showed that the treated lice had silicone present over their whole surface, as we expected. But it also showed that the silicone had formed a coating or plug inside the spiracles. Here, we have superimposed one of our actual X-ray pictures of silicone deposits in the spiracle onto an anatomical diagram, showing that Hedrin penetrated not only into the spiracle, but also into the trachea. This degree of penetration is extremely important because of the way head lice manage their water balance. Head lice do not produce urine, but excrete their water through their spiracles. Hedrin blocked this release route for the water, leading to the death of the louse.
The effect of Hedrin on lice was almost instant. Within 30 seconds they froze into immobility. This is their natural reaction when immersed in water, but lice do recover from water immersion. However, the Hedrin-coated lice entered a moribund state and never moved again. Their death was certain, but if they had recently fed, the results were even more dramatic.
If a Hedrin-treated louse had recently taken a blood meal, its gut actually ruptured due to the silicone coating dramatically reducing its ability to excrete water.
Hedrin lotion uses a unique patented silicone formulation with a very high molecular weight of 100,000 centistokes, particularly suited to causing a water vapour barrier and chosen because it proved the most effective. Hedrin's high molecular weight combined with its unique blend of silicones gives Hedrin maximum effectiveness against lice. This latest study supports previous trials that demonstrated the clinical performance of Hedrin, and an in vivo trial carried out in early 2008 showed that Hedrin has a 97% efficacy. To summarise the findings of the study, Hedrin does not appear to cause death by asphyxiation. Silicon from Hedrin penetrated into louse trachea and spiracles. Hedrin treatment triggered a fatal immobility reflex. Lice with blood-filled guts showed gut rupture after treatment. And Hedrin's unique patented formulation with high molecular weight silicone delivered maximum effectiveness. For full details of this study and other information on Hedrin lotion, please visit our website.